All right. Welcome, everybody. This is one of the few lectures every year that the CTSI presents for a distinguished speaker. So it's a treat for everybody to hear from these experts around the world. And today it's my honor to welcome Oscar Marín to be the distinguished speaker for this CTSI lecture. Oscar Marín is a native of Madrid, and he and I were raised around the same time in the 70s and 80s in Madrid. So it's funny talking to him because now I live in the United States, and it's rare that I get to hear slang from my generation, and it's just super funny. Plus, Oscar is a very funny guy. So it's been great to get to know him. I only met him a few years ago. But in the last few years, obviously I've realized I have a lot of things in common scientifically with him, but also just in terms of personalities, and we've become fast friends. And it's just getting easier and easier to just hang out with him. He did his PhD at the Universidad Complutense in Madrid, which is a very large public university in Madrid. And then after that, he came to the United States for a postdoc at UCSF in John Rubinstein's lab, where he did really well, so well that he received Young Investigator Awards from Europe and was recruited to establish his own lab in the Institute of Neurosciences in Alicante in Spain. And he was there until 2014, and then was recruited to be a professor at King's College, but he is the director of the Center for Neurodevelopmental Disorders of the Medical Research Council, which is a big deal, the MRC, as you all know. And his research has been in cortical development and with a focus on GABAergic interneurons. And I think it's probably well known that he is one of the leaders, one of the few best neuroscientists in this space of cortical development. But if you think about the techniques that he uses in the lab and the ease with which he incorporates in vivo calcium imaging or RNA sequencing, the use of DREADS, optogenetics to his lab and his projects, I'd say he's probably his papers are probably the most exciting, certainly in the last 5 years, but probably in the last decade that we've seen in developmental neuroscience. He is someone with enormous bandwidth. When you look at his CV and you pay attention to all the leadership positions that he has, it's really quite striking. He is a member of the Neuroscience and Mental Health Board for the Medical Research Council. He's in the editorial board of Science, Neuron, Development, Current Opinion Neurobiology, and several others. He's been in advisory boards, scientific advisory boards for numerous grant agencies, for pharmaceutical companies, private foundations. And of course, for his research, he's been awarded probably the most prestigious awards in Europe, including the EMBO Young Investigator Prize, the FENS, which is the Federation of European Neuroscience Societies Young Investigator Prize, and then more recently in Spain, the Ramón y Cajal Medal from the Spanish Royal Academy of Sciences. These are obviously the biggest awards in neuroscience in Europe. He's frequently invited to give lectures for the societies of neuroscience in countries like Spain, Portugal, Hungary, etc. I'm sure that he's most proud of the fact that he mentors a lot of grad students and postdocs. A very high percentage of whom end up going into their own faculty positions in academia and run their own labs. You may have heard of some of these names: Guillermina López Bendito, Víctor Borrell, Nathalie Deorter, and Isabel del Pino, among many others. So today, rather than speaking about his favorite thing, which would be the last few papers that he's published, he's going to give you an overview of his career over the last decade, at least, because the goal of these lectures is to be more general and appeal to a broader audience. And I think you'll be impressed and mesmerized. Having said all these really nice things about Oscar, I have to say that we could probably never be best friends, because being raised in Madrid, I'm a fan of Atletico Madrid, and he is a fan of Real Madrid. And so as he's giving his talk, I'm going to try to follow the Champions League match that is currently happening and make silly faces at him every time the other team scores. Anyway, welcome, Oscar.
Thank you so much, Carlos. That was probably the best introduction that I ever had. I should have recorded it. Now, you do realize that that we are really good friends, but not, super good friends, because he managed to schedule the talk right at the time of, one of the most important matches of football in the whole season so far. So thank you, Carlos. Right, so as Carlos said, hope not to bore you over the next 45 minutes or so. I prepared kind of a different talk of what I normally would have done, and it's going to be slightly more conceptual and a bit more thin on data, but hopefully you'll enjoy some of the concept. And I just want to touch upon one specific concept that is very recurrent in our field, but because of the venue and where I'm giving this talk, I want to give you an introduction and start with essentially a fairly easy to understand concept, and it's this idea that most psychiatric conditions that you can see have a very early onset in life, and obviously that varies over time. But as you can see here, most of the onset of many of the conditions that worry us in the clinical space do actually debut very early in life, and it's no surprise to anybody that this absolutely overlaps massively with the period in which we are still developing the hardware of our brain, our conscious, and where essentially all these processes are really happening. I think we tend to think about brain development as something that happens perhaps in utero or in early life, but we humans, if I were to identify the one feature that I think is unique about us is this ability to delay brain development over almost a couple of decades, which allow us to essentially model our hardware, at least a big chunk of our hardware, the cerebral cortex, to the environment around us and what we are learning during these, very early life events. And that actually determines to what extent, to a large extent, how we then react later in life. Both the very good positive things that happens to us in our infancy, and of course also what happens when it's traumatic and is not so good, that will also have a very major effect on the way we behave later in life. If I were to summarize what I think has been the most significant advance that I think in these conditions over the last probably 50 years or so, is the advance in genetics. I think that genetics has actually come to revolutionize the way that we understand these conditions, and there's a large number of people, many also here at UCLA and across the world, that have identified many genes now that we know confer clear susceptibility to developing these conditions. A lot of this risk is actually on the tail of these distributions for both conditions, so relatively minor effects, cumulative on a number of different variants. But it's also clear that, for example, both in autism spectrum disorders and in psychotic disorders, there is a large number of genes which, much higher incidence in the condition that can be attributed to actually changes into single genes. And this is, more clear for very early onset conditions like autism, but it's becoming also quite clear that even for late onset psychotic disorders, there are some genes which can be very deleteriously affected and predisposed to develop these disorders in increasing risk. So with these advances in genetics, I guess the challenge now, and again this is a very simple idea, is that we need to be able to connect what the genes actually do, and the emerging behavior sometimes many years later in life. And I guess the point I'm trying to make, that genes don't work in the outer space. They actually work to build the brain. So really understanding these conditions needs to go through the process of understanding how these genes contribute to building our brain. And as I was saying, this is part of the large interest that we have in the group, and probably across our center in London, is the idea that cortical circuits not only underlie some of most complex definitive behaviors of human beings, but also are known to be affected, disrupted in many of these conditions. Most strongly evident link into early onset epilepsy, but also autism and all the way to psychotic disorders. So understanding how risk genes influence cortical development, ideally in humans in this context, it is, in my opinion, the route to understand how these conditions come about and what we can do to actually treat them. And, for example, work from Carlos here on Fragile X illustrate the way, the power of trying to understand how brain circuits are affected illustrates this point. Now, I probably don't need this, but just in case somebody is not so familiar with the cortex contains two main classes of neurons, very well known. Pyramidal cells are known with this name because of the shape of the soma. They are excitatory neurons. They tend to depolarize their targets. They use glutamate, and they actually are the most characteristic cells in the cortex. They make most of the connections that you see in the cortex. They connect within a layer, across layers in a cortical column. They connect different cortical areas. They connect across the hemispheres and send projections down to all the way to spinal cord. So they actually make most of the action that's happening in the cortex. Now, the other population of neurons in the cortex, GABAergic interneurons, they tend to be hyperpolarized and they tend to inhibit their targets. They use GABA as main neurotransmitter. They are a lot more diverse from a morphological point of view. Not that pyramidal cells are not diverse, but GABAergic cells are very evidently diverse because they adopt very different morphologies. And in contrast to pyramidal cells, they're specialized essentially on making this local connectivity, making local connections in micro circuits. Now, one way to look at this is, and I tend to avoid symbols, but you can use the symbol of an orchestra. I can bring Gustavo Dudamel, your conductor for the LA Philharmonica, to say that interneurons somehow are a bit like the conductors of the brain. They synchronize activity of all the musicians. They synchronize activity of pyramidal cells, so they essentially know when they have to play or not play, and they are able to play with a single voice as opposed to do it in a completely asynchronous way. So this idea that interneurons are there essentially to allow synchronization of pyramidal cell activity is critical in the field. And that has led actually to another fairly heterogeneous concept I'm going to come back to in a second. And I think the idea that interneurons just plays this kind of role has taken a little bit of a life in the entire field. And as you will see in a second, that dominates the way that we think about problems in the cortex. And I think that's right and it's fine, but as Megan said, typically simple solutions for complex problems are a bad recipe. Now I want to focus my talk today around one very simple concept that is pervasive in hundreds of papers dealing with cortical function and with neuropsychiatric conditions, and this idea that somehow the balance between excitation and inhibition is perturbed in these conditions, and that this actually underlies pathology. And I think that conceptually this is a very simple idea that you can represent with a kind of neat image that you can get from the internet. But when you get to the nitty gritty of this, it's really difficult to come to understand what we actually mean by this idea. And actually my feeling is that that maybe the conclusion of the talk is that EI balance cannot be disrupted in the cortex. It can be pathological, it cannot be disrupted. If you disrupt EI balance, you essentially have seizures and then you eventually die unless you control this. So this whole idea that EI imbalance is the underlying cause of everything we see wrong in the cortex needs to be modulated by, sinus specificity. So today I want to give you examples of some of our work in around two general concepts. And I think that, my take-home message is that it is very difficult to understand dynamics process in a developing system, because essentially everything is trying to, react to, adjust to the changes that you introduce initially. In a developing system, in the dynamic system, everything that you do initially will have cascading consequences. I'll show you in a second. And all those, consequences are intended initially to restore this idea of EI balance, to, to try to bring cortical networks into some sort of balance. Now, this idea of EI balance can be seen as at two different levels. I'll give you samples of those. On the one hand, you can have you can take a population view of this problem and think about this in the way you build cortical circuits. You need to take into account how many of these neurons you put together, how many excitatory cells, how many inhibitory cells you put together, and there has to be some quantitative aspect into this equation, and it should matter actually how you build these circuits. The second is a slightly more kind of physiological take on this, is that for every single neuron excitatory inhibitory, it will need to maintain more or less baseline firing activity within a normal range, and this has to be essentially checked over time to be able to keep it on this kind of functional range. So there's a population level of activity that is being controlled and balanced, and individual neurons themselves have their own mechanisms to control this balance between excitatory and inhibitory activity. So let me tell you a little bit about the first, and this actually comes from a relatively general observation, which I think it's in my opinion, fairly puzzling, is that if you look at the entire mammalian radiation, and you see cerebral cortices of very different sizes and shapes, one thing that is relatively common is that the ratio between excitatory and inhibitory cells across cortical in different species, that the range, as I said there, sometimes up to 1,000-fold difference in size. It is quite narrow, and this is relatively recent data over the last few years. We now know that there are probably about twice as many interneurons in the cortex than of humans, and probably also primates than in the mouse. But it's still, this fraction of cells, seems to be quite narrow. So there has to be a system that has evolved to maintain this narrow kind of bandwidth in which you correlate excitatory and inhibitory cells. And obviously, you can think about this in different ways. You can think about mechanisms that make coevolution of the progenitor pools at the same time, so that when you grow your cortical regions and you have many more pyramidal cells, in parallel you expand the pool of progenitor cells for interneurons. But the second mechanism, which we think is even more important than that, is that you can actually overproduce neurons, as you do through the entire nervous system, and then you control final ratios by adjusting the number of cells that you actually kill during postnatal development. And we've been very interested in looking at this over the last few years, and it's actually following very early work from Arturo Alberto-Illa at UCSF, described that interneurons undergo very heavy programmed cell death in early postnatal development. Again, this is not a novel idea. I think programmed cell death happens across the entire nervous system. It's just that in the cerebral cortex, and in the case of interneurons, it becomes really very dramatic. And this is actually work from two former postdocs in the lab, now gone and on their own independent operations. And what is interesting is that this process of correcting the final numbers of cortical neurons happens during early postnatal development in the mouse. And what is interesting is that it happens in two more or less consecutive phases. In green, you have a first period of programmed cell death for pyramidal cells. This is the relative numbers when the animals are born. And you can see over a period of about 3 days or so, they correct, approximately about 10% of the pyramidal cells die. And this happens in an heterogeneous way through the cortex. I'll come back to that later. And then immediately, following this process of pyramidal cell death, there is a new wave of programmed cell death that now affects GABAergic interneurons. And work that we have done over the last few years have actually shown that these two processes are extremely interlinked. I'll show you some evidence about this. To the point that we think that actually interneurons are always going to be following what pyramidal cells are dictating, and this happens all the way to physiology later on. But even at these earlier stages, when the neurons are just integrated into cortical circuits, their final numbers are going to be immediately adjusted based on what pyramidal cells have done before. Now, this cartoon summarizes what we think is actually happening, that pyramidal cells correct their numbers, and then interneurons follow and correct and stabilize based on the numbers of pyramidal cells that finish. What is interesting is that during development, it seems that interneurons are actually programmed to die. So if you take interneurons and you put them in culture on their own, they will all undergo programmed cell death. So they have an internal clock. They have a mechanism through which they will undergo programmed cell death. They will be eliminated unless they are rescued by their connections with pyramidal cells. So it's this early phase of connectivity between pyramidal cells and interneurons that actually make them to survive past this window, which again in mice is only about 5 days. So it's a very defined window of time in which these numbers are corrected in early postnatal development. I'll show you some evidence that we think that the mechanism that drive programmed cell death in interneurons is a very strong increase in this phosphatase P10 that actually abolishes AKT signaling and drives cells to apoptosis. So what pyramidal cells connected to interneurons do is essentially maintain low P10 levels so that the cells that are connected can survive. So it's an interesting kind of checkpoint, a rise in P10 levels, and unless you have a mechanism to prevent that from happening, then you will be gone from the cortex. Let me show you two pieces of data to support this idea. One is that you can abolish neurotransmitter release from pyramidal cells in the early postnatal cortex, for example, doing genetic experiments in which you remove bGlut1, bGlut2. These two transporters are required for glutamate to be loaded into vesicles. So if you don't have these transporters, that essentially pyramidal cells will be kind of fighting blanks. They will not be able to release glutamate. And if you do this in early development, this is just for a population of interneurons. What you see is that many fewer interneurons survive this process. So you really need the connectivity, the glutamatergic neurotransmission for these cells to survive. Now, if you do these manipulations after the window of programmed cell that happens, there's a number of adaptations. There will be synaptic changes and so on, but they will not interfere with the actual number of interneurons. So this is kind of a checkpoint in early postnatal development in which the number are adjusted over a few days. Now, I was telling you about P10, and this you can see in normal tissue is that at some point some interneurons get very, very strong levels at this point. If you do calcium imaging on these cells, you will see that the cells that have very high P10 levels have very low levels of activity. And if you come back, if you're doing chronic experiments, you come back the next day, these two cells will stay in the cortex. This cell will be gone. It will be a hole in that place. And what is interesting is that if you remove P10 from postmitotic interneurons, so you do a genetic experiment and you take away this phosphatase, then all the interneurons now survive. So if you take away the mechanism, the molecular mechanism through which these cells are normally discarded, then all interneurons will survive. And this is one, very silly example of how, complicated understanding developmental processes, because P10 is mutations in P10, loss of function mutations in P10 is being associated with autism. It's actually one of these cases of macrocephaly in autism that is very well understood. And because P10, counterbalance AKT, it's always been thought that, what happens in these kids is that they actually make too many neurons, which actually may be happening in this case. But what these very simple experiments also show, and we're trying to look at this in human neurons, is that perhaps part of the problem is that you don't kill enough neurons, not just that you make a lot, but you may actually not kill those that are poorly connected because you don't have the mechanism, the molecular mechanism through which you normally eliminate these cells. And then you get, you end up having circuits in which you have neurons that are not properly integrated. And I'll come back to that in a minute. I'm not going to show you experiments, but what is really fascinating is that this whole system is set up to modulate the number of interneurons based on the activity of the cortex in these early phases. So going back to this idea that interneurons are there to make sure that pyramidal cells have the right levels of activity, and that you can actually sustain that over time. And you can do experiments, for example, with dreads in this window of time, and you can not just remove glutamate altogether, but maybe just decrease the levels of activity of pyramidal cells, and then you will end up with the cortex with fewer GABAergic interneurons, even though the numbers of pyramidal cells will be normal. Or the other way around, you can actually use dreads to activate pyramidal cells in that window of time, and then your cortex will end up with many more interneurons than normal. So the system is set up to adjust the final numbers of interneurons to the numbers of pyramidal cells and their activity during development. Now, one prediction that we could make very early on with this system, when we have all these alterations and seeing how interneurons clearly follow pyramidal cells, that you could do kind of a very radical experiment in which you prevent programmed cell death from pyramidal cells, so they will not die. And then the prediction will be is that you escape this window altogether, then now none of the interneurons may actually die because you may have many more pyramidal cells. Now, the nice thing about this is that you can actually do that experiment because we know that two mitochondrial proteins, BAX and BAC, are absolutely required for apoptotic cell death. So we did experiments in which we did this complicated crossing to get these mice, in which essentially you prevent only pyramidal cells. So this is only happening in pyramidal cells. You prevent them from dying. And now, as opposed to having 10% less pyramidal cells, you end up with animals that has the same number of pyramidal cells at the time that they were born in. And these animals are a bit microcephalic, not much, but they have many more. Now, when you go and look at the interneurons now, and so you just one example of these pyroglobulin fast spiking cells. Now the interneurons, are rescued, so none of the interneurons actually die. You can actually rescue the whole number, so you essentially end up with very little cell death when you have these mice and this particular condition in the somatosensory cortex. So that brings this idea that the final site architecture of cortical regions is somehow dictated in this very early phase of postnatal development, in which the ratio of these populations of cells get adjusted. Now, you might be thinking at this point, does this actually matter? Is that really important that you end up with more or less GABAergic interneurons? So when we did these experiments, we, as usual for all of us, we have our favorite cortical region, which typically is the one that we have a functional assay for it, or the one that we've been looking for the past 20 years, and that's the one that we look at. For whatever reason, we did these experiments in somatosensory cortex, and we thought, well, this is quite neat. And at some point, Ellie joined the lab, and she wanted to look at this a bit more broadly. And a project that I started by just exploring something like that has taken a life, and now a few years later, we're still trying to wrap this up. But this image illustrates the fact that GABAergic interneurons, the density of these cells, and this is for total number of GABAergic interneurons, is extremely heterogeneous across the entire cortex. It's by no means the same kind of configuration of interneurons. And as I said, this is just total numbers, but even if you look at cell type specific distribution, the heterogeneity is astonishing. So there are no two cortical regions that actually have the same complement. They have the same complement in the sense of having the same quality of interneurons, but the quantities, the proportions are completely distinct. And I think that is going to be an interesting thing to explore in the future. Now, we think that part of this is because cell death is not homogeneously controlled across the entire cortex, and that actually kind of makes sense because, by definition, every cortical region, based on Brodmann work from many years ago, by definition, every cortical region will have a complement of pyramidal cells, excitatory cells, and connectivity, and so on, that will make that region special. And if interneurons are following pyramidal cells, then it kind of calls to reason that that particular cortical region also have a special complement of GABAergic interneurons. And we still don't understand quite, the entire logic of this process, but we have come up with experiments in which we perturb the death of pyramidal cells, and we create these kind of Frankenstein mice in which the relative proportions of GABAergic interneurons are differentially affected in in different regions of the cortex. And I'm going to tell you now through a few experiments that we haven't published yet, and you will see the naivety of these experiments. But I just want to make the point that even if we haven't managed to get this right yet, I think it points to, it gets to the point that the way these different cortical areas behave is highly influenced by the final ratios of GABAergic interneurons and circulatory cells that you have. So we have these funny mice in which we accidentally found that visual cortex, which in humans is also a particular region. In primary visual cortex in mice, there's essentially no cell death, or if you want, there's no cell death that we can detect, below the levels of, well, 3 or 4% difference that we can actually statistically see in a group of animals. So for whatever reason, that region has very little programmed cell death. So if you prevent the programmed cell death of pyramidal cells, nothing happens. Actually, you end up with more or less the same number of pyramidal cells, same numbers of interneurons. In the somatosensory cortex, which is the place where I told you, we did experiments originally, you have, very macrocephalic cortex in which you have many more pyramidal cells. You prevent the cell death of these cells, and you end up with a lot more interneurons. And in the medial prefrontal cortex, something strange happens, and we now have evidence why this is happening. There is no match of programmed cell death for pyramidal cells, and yet you end up with a great number of many more interneurons, primarily actually somatostatin GABAergic interneurons. And we think that this is the case because these cells are rescued from dying not by local pyramidal cells, but actually by excitatory connections coming from other cortical regions. So you end up with this kind of mosaic animal that it's, in which the ratios are more or less altered in different ways. And you can think, well, you have more or less a controlled cortical region, and then regions in which we have changed the ratios in different ways. Now, I'm not showing you what's happening in the other 23 cortical regions, and that's why, obviously this approach to looking at this problem is very naive. But I just want you to take you through some experiments that illustrate some of the points I want to make. And this actually worked from student Risto and a postdoc in the lab, Marín. So they decided to look at how these animals behave, and what they wanted to see is whether they can identify a way to test more or less the same thing across these different cortical areas. And we were actually inspired by work from Carlos' lab, and we set up this go-no-go task in which the animals need to discriminate between screens with bars of different orientations, and which they need to learn a rewarded grading and a non-rewarding grading. So a relatively straightforward, easy task. And then we set up more or less the same task now for the whisker system, in which now instead of presenting visual stimulus, we presented them with sandpaper of different grains. And one of the grain, the coarse grains will be rewarded, and the fine grain will not be rewarded. But essentially, it's the same structure of the task. And then in the final set of experiments, trying to get in the same context, trying to get a prefrontal function, we did something that has been primarily done in rats and in monkeys, which is try to do sequential learning. And the idea here is that mice and monkeys and humans too will learn first one of these tasks, and then they will be able to generalize the rule that they are applying, in this case, a go-no-go task, so that learning the second visual, in this case, the visual stimulus, will be much faster than in the first task. And some people call it that transfer learning or learning to learn sort of task. So we take the animals through this task, and our visual cortex was kind of our control set of experiments because we would not expect to see many differences there. And the reality is that we don't. I mean, control and mutant mice learn more or less at the same rate. And if once the animals have learned, if you take them to psychometric experiments, trying to make the task more difficult, the curves are indistinguishable between control and mutant mice. Now we take this, a different set of animals to the whisker task, and here we were a bit surprised because these animals actually end up with about 10% more pyramidal cells, about 35% more GABAergic interneurons in the somatosensory cortex. We know that both of these tasks, this task is absolutely dependent on V1, this task is absolutely dependent on S1. So you mess up, the ratios and the number of cells, across this region, and these animals have no problem learning these tasks whatsoever at exactly the same rate. And once the animals have learned, if you do psychometrics, again, their ability to perceive different stimulus is identical. Now, what's interesting that these animals that manage to learn no problem the visual discrimination task and the tested discrimination task struggle with sequential learning. And in the case of control animals, as I hope you can see here, you can compare this graph to this graph. So here, on average, it takes between 3 and 4 days for the animals to learn, and learning here established here at this dotted line. Here they're above 85% success. What you can see is that in this sequential task, now the animals that have learned before the whisker task and now are taken through the visual task, they can learn much faster than control animals that are naive to the task, and they typically learn in about 2 days or so. But interestingly enough, these mutant mice in which prefrontal cortex ratios are changed, even though they can actually learn at normal rates in naive conditions, they cannot transfer the learning. They fail to benefit from having learned the whisker task in the previous week. So that tells you 2 things. One is that you can really screw up the cortex quite badly and change a massive number of proportions in a non very sophisticated way, and the animal will still find a way around the task and do pretty well. And I guess the other thing that you can learn is that everything has a limit, and there are things when you start pushing that are slightly more delicate, and perhaps that requires a bit more balance. Let me show you another piece of data that at least can lead to begin to understand why this might be happening. So we've been very interested in trying to understand what changes in the prefrontal cortex when these animals are going through these tasks, and primarily through this period of time in which the control animals are learning much faster, whereas the mutant mice are still moving in the normal phase. And we record from the prefrontal cortex, and we did analysis in which we separate our units, most of them pyramidal cells, although we have looked at fast spiking cells too, into those that are selective for the go stimulus, for the eventually rewarded stimulus, and those that are selective for the no-go stimulus. And here, this is the responses to to both, and these are go selective cells because, as you can see, their firing increases more for the rewarded stimulus here in yellowish brownish than the no selective stimulus, which is here in blue. They will respond to both, but much higher for the rewarded stimulus, and that actually doesn't change much between control and mutants. And in a very naive way, you can say, well, this might actually drive behavior in such a way. Now, what's interesting is that for no-go selective cells in the prefrontal cortex, same place in both animals. In control animals, the no-go selective cells still fire more than for the go stimulus, but you can see that essentially no-go signal. What essentially they do is they suppress firing on the presentation of the stimulus. Now, what's interesting is that in the mutants, for whatever reason, and the only proxy we have is that we have changed actually the number of interneurons in that cortical region. You see now that no-go selective cells also fire on presentation of the stimulus, which may actually kind of fit with the idea that the animals have not learned this early phase of the task. Let me wrap up this part. And again, I don't want to go down real on the data, but I think there are several very general conclusions that I think are interesting. One is that mammals have evolved a fairly robust system to coordinate the numbers of GABAergic interneurons and pyramidal cells, and the ratios are adjusted in this early postnatal development. I think the second idea, which we didn't predict, is that this process is extremely heterogeneous across cortical regions, and that actually contributes to each cortical region having a slightly different complement of GABAergic interneurons. And until now, I think we have looked at this much more from the perspective of qualitative aspects, whether they have the same interneurons in different places. And I think that at some point we're going to need to start thinking about quantitative aspects and how much influence it has that a particular cortical region has twice as many VIP interneurons than another region. And the final bit, and I know that this is still a very thin terrain, and I guess we need to find better ways of going about this, is that in spite of these very dramatic changes, the cortex always, try to manage to find a way around a particular task and try to find ways to compensate for fairly dramatic changes. And, I haven't shown you any data on this, but if you look at baseline firing, for example, in some of these animals in which you have twice as many interneurons as you should, baseline firing for pyramidal cells have not changed. So the cells have arrived to a point in which they can actually control their firing properties, in spite of having, many more GABAergic interneurons around. So again, going back to this idea of AI balance, just basic, properties, firing rates, for example, might not be a really good predictor of something going wrong, and we may need to find a different way of interrogate these computations. Right, let me change gears for the last bit of the talk and tell you something more from a cell-centric kind of perspective. And this is kind of related work looking at essentially the function of power-booming, fast-spiking basket cells. And this idea that, this slide just conceptualized this idea that different classes of interneurons, because of their intrinsic properties and their connectivity, they are able to help synchronize in cortical networks at different rhythms. And, for example, PB basket cells have been demonstrated to be critical to synchronize activity in the gamma rates, these, really fast oscillations that are known to be extremely important for cognitive function, and it's been strongly related to, for example, working memory processes. So one thing that is quite interesting is that this is perhaps one of the best clinical links to, cognitive deficits in people with schizophrenia and even naive patients. We know that, for normal individuals, cognitive tasks that involve a working memory process requires gamma synchronization, for example, in the prefrontal cortex. And this is something that is very clearly impaired in people with psychosis that has cognitive deficits, their inability to synchronize on the gamma rhythm. And that has actually suggested that, people with psychosis, primarily with schizophrenia, would have some sort of deficit in the function of GABAergic interneurons. There is a little bit of pathological evidence of this, primarily from postmortem studies. There's a lot of work from David Lewis, for example, in Pittsburgh, suggesting that many GABAergic markers are decreased. Expression of power-booming in these cells is decreased. The number of synapses made by PB basket cells might also be decreased in these people. So there's good evidence from physiology pointing out to gamma rhythm deficits. There's good evidence from neuropathology suggesting that PB cells might be disrupted. So a number of years ago, we wanted to see whether we could actually model this in mice and come up with a very simple model that disrupting interneuron development, probably through some sort of genetic alteration, will lead to abnormal gamma rhythms in the cortex and will be the basis for cognitive deficits in these cases. And this actually worked from 2 former postdocs in the lab. And our entry point to this story is not really a super strong candidate for schizophrenia genetics, although RB4, which is a tyrosine kinase receptor that we've been studying for more than a decade now, has been linked actually to different conditions. It's been some strong loss of function mutations have been linked to both psychosis and intellectual disability. There's more recent data from the last GWAS study suggesting also that more general variation in this locus also correlates with psychosis. And as I said, this is tyrosine kinase receptor, and we've been studying the function of this receptor for many years now. Now, what is interesting about this, and we found this initially in mice, but that this has been replicated subsequently in monkeys and in humans, is that RB4 is exclusively expressed in the postsynaptic side of soma and dendrites of power-booming fast-spiking interneurons that are contacted by synapses coming from pyramidal cells. So it's present on interneurons. It's not expressed in pyramidal cells. It's only present in interneurons, but it's always on the receiving end, so to speak, of synapses coming from excitatory cells. And what we have found over many years is that this tyrosine kinase receptor is critical for the building of these synapses. It recruits a huge number of proteins into the membrane, and allows actually the formation of these excitatory synapses onto GABAergic interneurons. And incidentally, many of the proteins that is responsible for recruiting and being expressed at these synapses have also been linked to many conditions, particularly autism spectrum disorders. So it seems to us that these synapse in particular it's strongly connected to many of these conditions. So we did create conditional mouse mutants for RB4, in which we actually remove RB4 only from fast spiking cells and actually also from somatostatin cells. In somatostatin cells, it's not expressed, so it actually doesn't do anything. And in these mutants, what we found is that in the absence of RB4, there is about a 30% decrease of synapses coming from pyramidal cells into parvalbumin interneurons, and that's essentially the core of the phenotype. Now, these developmental defects, so the absence of about 1/3 of the synapses made by pyramidal cells into fast spiking interneurons, leads initially to reduced activity of parvalbumin interneurons. You recruit a lot less GABAergic activity, by the deficits on GABAergic on excitatory synapses. Now, what's interesting is that over time, these reduced number of synapses first lead to reduced inhibition, and therefore increase pyramidal cell activity, and eventually this increased pyramidal cell activity, so hyperactivity in the cortex, lead also to increased activity in fast spiking interneurons. So something that starts as a very prominent deficit in the number of synapses initially cascades down as ending in a cortex that is hyperactive, in which not only pyramidal cells are hyperactive, but also dysfunctional fast spiking interneurons are hyperactive. What's interesting is that in these mice, compared to control mice, there's heavily, strongly increased gamma redoin in these mutants. So the fact that the animals end up having more pyramidal cell activity that drives more PV activity, end up with very abnormal gamma range activity, noisy gamma range activity in the cortex. And that's quite interesting because naive patients that have not received medication, for example, they also have very strong abnormal increase in gamma readings in first episode psychosis. Let me finish with this final thing. So again, going back to the idea of compensations in the cortex. These experiments in mice allow you to have a very tight control over what you are actually altering, and obviously in patients, mutations, for example, will affect more broadly. But in this case, the one thing that you can see is that that very small changes in this case about 1/3 of synapses contacting a very specific population of GABAergic interneurons cascade into major problems across the entire cortex. Now, there are two things that are quite surprising in these experiments, but maybe not so surprising. One is that when you look at pyramidal cells, which we haven't touched, they are unaffected initially by the genetic effect. They have a very prominent decrease in synaptic botons, and that is consistent with the idea that after hyperactivity in the cortex, these pyramidal cells will start to prune connections among them. And this is interesting because this is one of the features of pathology in psychosis. Layer 2-3 cells, for example, in the human cortex tend to have a lot less spines than normally. So I'm not saying that the cause of that problem in humans is that you have initially a problem in interneurons that cascade down into pyramidal cells. What I said is that all these things are always interconnected, and sometimes you may actually start perhaps with a problem in pyramidal cells that will cascade and impact the function of GABAergic interneurons. And in this case, it's the reverse. You start with a problem in GABAergic cells, and then you end up causing a major phenotype on pyramidal cells. Now, the second thing that we are exploring at the moment, which is quite interesting, is that in these mutants, you are only affecting parvalbumin interneurons, or the connectivity of the parvalbumin interneurons in the cerebral cortex. We're not touching anything else in these mice. What we see is that over time, these mutants develop hyperactivity. And if you do microdialysis experiments, you see that there is massive dopamine increase release in the striatum. And this is D1, D2 dependent. and actually reproduces what you normally observe also in patients with psychosis and schizophrenia that end up having striatal hyperdopaminergia, a strong abnormal release of dopamine that correlates with psychosis in a large fraction of patients. And again, this is something that we're exploring at the moment. This is caused by abnormal increase in the activity of dopamine neurons in the midbrain, which are completely unaffected by this mutation. This mutation is only affecting GABA cells in the cerebral cortex. And yet, cascading down for that hyperactivity in the cortex, weeks later, you start having problems in the midbrain, and these cells begin to fire more and start releasing more dopamine in striatum, and the animals end up becoming hyperactive. So again, that actually suggests that these cascading problems define different phases of a particular condition. In the case of psychosis, you may define a prodromal phase in which you primarily have negative symptoms and cognitive deficits before the onset of psychosis, all the positive symptoms that comes along with that. Suggesting again that understanding the disease process, the evolution of these cascading deficits may actually give us a way to intervene much earlier than what we do at the moment. Right. So in this second part of the talk, wrapping up now, I told you something that we find quite interesting. And we have more and more data suggesting that deficits in GABAergic interneurons, particularly fast-spiking interneurons, are highly related to different conditions, and many genes that are critical for these conditions seems to be affecting the development of these interneurons in particular. That these early alterations that are, in principle, easy to define, synaptic problems, the formation of synapses cascade in ways that are sometimes difficult to predict. And then finally, that this impairment in cortical circuits may actually cascade away from the cortex and start impacting other systems that may actually account for some important symptoms in these conditions. That might be a bit blunt, but that's a summary of my talk. I think that it's going to be impossible to understand neuropsychiatric conditions unless we frame it in the context of this developmental process, the way the brain is organized, the way the cortex is organized in cascading ways. You need to understand what are the consequences through development, and you may actually need to be able to define what windows you're going to be able to intervene, and that might actually vary across the life of individuals. Let me finish here. I think I acknowledged everybody as I went through, but that's essentially where we work, in the shadow of the SAR, which is the tallest building in Europe, some of the most expensive real estate in Europe too. That explains the cost of our mice. And I'll stop here and take any questions you may have. Thank you.
What's your opinion about transcranial magnetic stimulation regarding deterring the changes or improving the condition?
I think it's still poorly understood how it works and how effective it might be in these conditions. But I think it's one of the ways in which we can actually interfere with the function of some of these cells.
Thank you, Oscar. So the learning rules, the algorithms that control the connectivity and synaptic strength generally operate at the synaptic level, not at the cellular level. Not always, but generally. So in your mutants in which you have more inhibitory neurons, one might imagine that you really have the same number of inhibitory synapses. Do you have an answer to that, if that's the case or not?
So we have looked at this at different stages, and it actually changed quite dramatically. So at the beginning, you see, over the first few weeks, you see a massive increase in the number of synapses, inhibitory synapses, for example, in the prefrontal cortex. When we look at the time that we are changing, we're seeing the behavioral changes, we don't see much difference, as if finally the number of synapses have normalized. So that seems to be compensated, and that kind of is in line with the idea that the baseline firing of these cells is not significantly different between mutants and controls.
The aerial difference in matching excitatory to inhibitory neuron is really striking. And I'm wondering, it could be just all driven by activity, tightly tuned, homeostatic, et cetera. But there also might be molecular cues in the environment, such as through overlapping populations of heterogeneous astrocytes or something like that. And in the years past, Dennis O'Leary tried to get at that by, transplantation experiments. I'm wondering, have you been able to look at perhaps set molecular cues in addition to activity?
Yeah, so we're really very actively looking into that at the moment. And the primary reason is that we did the experiment of abolishing completely GABAergic interneuron cell death as a way to say, well, if it's all driven by interactions that are activity dependent, if you prevent programmed cell death, then the hypothesis will be that you create GABAergic interneurons, you distribute them homogeneously, and then you should have more or less the same ratios across. And then if you take away programmed cell death, you should see more or less a tabula rasa of distributions. And that is not what we see. And that actually suggests that even though programmed cell death sculpt these final numbers, before programmed cell death, the cells are already deployed in a relatively heterogeneous way. And to me, that is good evidence that there has to be some sort of molecular mechanism, which we are trying to get our head around, that suggests that some regions capture or host more interneurons to start with than others, and then activity probably prune them into final configuration. I think it's not an easy problem to get around, because it's difficult to find very sharp boundaries, for example, in molecular cues. There's something that we have looked in the past, and during development, you don't typically tend to see very, alluded Dennis' work, for example. I think that's a good example of how difficult it is to find very clearly defined molecular cues. And in the distribution of numbers, what you see is sharp boundaries. You can use the interneurons to look exactly where the boundary between two regions are. It's as good as that, and if you have the right cell for that particular region. So we need to make these two things compatible: the molecular patterns that perhaps are not so sharp, and the final result. But we'll work on this over the next few years, I think.
Hi, great talk. This might be a bit more of a general question, but are abnormal gamma oscillations also associated with other neurodevelopmental disorders as well?
Yes, I don't think it's been studied as careful as it's been studied in psychosis, but there is some evidence of abnormal gamma rhythms also in some syndromic autism conditions. Yes.
Okay, thank you.
I had a follow-up question to that, since it relates to the ERB4 experiments. So you showed some deficits behaviorally in locomotion, and obviously some things related to dopamine, which are important in the context of schizophrenia. But I'm curious whether you've gone back to that experiment and looked at, behavioral assays like the visual discrimination, go, no go, because would you expect differences based on just that slight reduction in synapses onto PVs?
Yeah, that's a good question. We haven't revisited that, and maybe we should look at this more carefully. I mean, one of the things that is quite interesting, the fact that you have a 30% reduction in synapses is always puzzling. I guess that why don't you lose all the synapses, I guess, or why do you have a more strong phenotype? And we always tend to think that this is because there's probably another molecule that compensates and helps and do this and that. One of the things that we have learned more recently is that actually ERB4 is not expressed in every glutamaterase synapses contacting PV cells. It's actually expressed only in about 40% of the synapses. We don't know what are those. We don't know whether they are from local cells, from different layers, or what are those synapses. But one possibility is that perhaps those synapses are somehow different. We know, I went through it very quickly, but we know that local protein synthesis is very important for the making of these synapses. And perhaps that is more connected to plasticity. So perhaps those synapses are particularly plastic and are the ones that we see that typically are corrected or not. So maybe in the type of behavioral experiments you're suggesting, maybe we will see a deficit in particular for this. So we need to do that.
Istvan Modi was listening to your talk online, and he's asking whether ERB4 mutations, which are present in numerous forms of cancer, are these mutations present in PD interneurons as well? Or I guess maybe he means in neurodevelopmental conditions as well. Are there similarities in the mutations?
I think so. I think at least some of the mutations have been described across and that are tumorigenic. I think that they've been linked to intellectual disability problems, and we don't think that they interfere with the generation of neurons. But there might be evidence that it might be important for migration as well as for synaptogenesis. And again, I think the genetics are, I think that less work out for other genes, but I think there are overlapping mutations.
Any other questions? Great. Well, then join me in thanking for a great talk.